Hello everyone, this is lecture 7 and in this lecture we will learn about enzyme inhibitors. So what are enzyme inhibitors? They are molecular agents that interfere with the catalysis of the enzymatic reactions by slowing it or halting it. Okay, and as you know, enzymes catalyze virtually all the cellular processes. So many enzyme inhibitors, they are used as important pharmaceutical agents, that is drug. Okay, and for example, aspirin, it's a famous drug which is used as a painkiller. Okay, so what aspirin do is actually a inhibitor. Okay, it inhibits the enzyme that catalyzes the first step in the synthesis of prostaglandins, and this prostaglandins, it involved in many processes, including some that produce pain. Okay, so that means as aspirin can actually stop the synthesis of prostaglandin, it can be used as a painkiller. Okay, and not only as drug, enzyme inhibitors are also important to study the mechanisms and enzymes, and also to understand different metabolic pathways. Okay, anyway, uh, the enzyme inhibitors, they are broadly classified into two groups: reversible inhibitors and irreversible inhibitors. Okay, and reversible inhibitors are three types: competitive, uncompetitive, non-competitive. And irreversible inhibitors, there are also three types: group specific, affinity labels, and suicide inhibitors. The reversible inhibitors are named so because the effect of the reversible inhibitors can be reversed. I mean, when we add the inhibitors, they slow down or stop the reaction rate. Okay, so this effect, that means the effect of the inhibitors on the reaction rate can be reversed. Okay, but in case of irreversible inhibitors, the effect on the reaction rate by these inhibitors cannot be reversed. Okay, so that's why they are called irreversible inhibitors. Okay, in this lecture in this chapter we'll learn about these inhibitors, and first we'll start with competitive inhibitors. Okay, so why they are named so? Because a competitive inhibitor competes with the substrate for the active side of an enzyme. Okay, so while the competitive inhibitors are there, they bind with the active side, so the substrate cannot bind with the enzyme. Okay, so thus the reaction cannot proceed. Okay, and as the competitive inhibitor, they can compete with the binding. Okay, they compete with the substrate to binding. So actually the structure of the competitive inhibitors, they are similar to the substrate. Okay, so that's why they can compete with the substrate for binding with the enzyme. Okay, now as the competitive inhibitor, they compete with the substrate for binding, the effect of this kind of inhibition can be reversed by increasing the substrate concentration. I mean, if we increase the substrate concentration, they can reverse, they will be more competitive with the in comparison with the inhibitor. Okay, so thus the effect means when competitive inhibitors were added, the reaction rate slows down or halted. Okay, so if we increase the concentration of substrate, then again the reaction rate will increase. Okay, one example of competitive inhibitor is methotrexate. Okay, and this inhibitor is a structural analog of tetrahydrofolate, which is a coenzyme for the enzyme dihydrofolate reductase, and this enzyme, it plays a role in the biosynthesis of purines and pyrimidates. That means this enzyme is involved in the cell growth, cell multiplication. Okay, and as the methotrexate, it act as an inhibitor for this enzyme. This inhibitors is used as a cancer drug to treat cancer. Okay, because we know in case of cancer, uncontrolled growth of the cells occurs. So these inhibitors can enzymes this enzyme. That means it can inhibit or interfere with the cell growth or cell proliferation. Okay, so that's why it can be used, it is used as a cancer drug. So this slide, it shows the overall ways by which the competitive inhibitor works. So competitive inhibitors, they bind with the free enzyme, okay, and thus inhibit the reaction. Okay, they don't bind with the enzyme substrate complex. Okay, and we already know that the competitive inhibitors, they are structurally similar with the substrate. Okay, that's why they can bind with the active site where the substrate bind. Okay, this slide shows another example of competitive inhibitor. Okay, so malonate is an inhibitor of this enzyme succinate dehydrogenase. Okay, and the succinate dehydrogenase, it's catalyze this reaction. It's produce fumarate from succinate. Okay, so if we add malonate in this reaction, malonate will bind with the enzyme, and this catalysis will not occur. Okay. Okay, and now about reaction. So as we add, as we know that the inhibitors, they interfere with the reaction rate. So whenever inhibitor will be added, the rate equation will be changed. Okay, and we already know about the rate equation, the Michaelis-Menten rate equation. Okay, which is this one: V0 equal to Vmax substrate concentration divided Km plus substrate concentration. So when an inhibitor is added, the reaction changes like this: Vmax substrate concentration divided alpha Km plus substrate. So a new parameters alpha come here. Okay, and the value of alpha is equal to 1 plus inhibitor concentration divided Km, and this is the linearized form of this equation. Okay, and if we plot them in the graph, they look like this. Okay, now uncompetitive inhibitor. Okay, so they are named so because uncompetitive inhibitor binds at a site which is distinct from the substrate binding site, and unlike a competitive inhibitor, binds only to the ES complex. Okay, so uncompetitive inhibitor don't binds with the free enzyme, rather they binds only with the enzyme substrate complex. Okay, and an uncompetitive inhibitor acts by decreasing the turnover number rather than by diminishing the proportion of enzyme molecules that are bound to substrate. Okay, so that means actually when the uncompetitive inhibitors, they bind with the enzyme not in the active site, okay, other than the active site, in a distinct site. Okay, so when the uncompetitive inhibitor binds with the enzyme, they can cause a structural distortion in the active site of the enzyme, thus is interfere with the catalysis. So as the active site of the enzyme is disrupted, they cannot catalyze the reaction. Okay, this is how that is how the competitive inhibitors work. Okay, so that means they don't interfere with the substrate binding, rather they interfere with the catalytic power of the enzyme. Okay, and as this enzyme, they don't interfere with the substrate binding, we cannot substrate binding. So basically increasing the concentration of substrate will not reverse the effect of uncompetitive inhibitors. Okay, which means the effects of uncompetitive inhibition on Vmax are not reversed by increasing the substrate concentration. Okay, and at low substrate concentration, that is when substrate concentration is much lower than the Km, the effect of uncompetitive inhibitor becomes negligible. Okay. This slide, it shows the ways by which uncompetitive inhibitor works, which you already know, that the uncompetitive inhibitors, they bind with the ES complex, not with the free enzyme. Okay, and as the uncompetitive inhibitors, they don't bind with the substrate binding site. Okay, so it's not necessary that they have to be structurally similar with the substrate. Okay, and once the these uncompetitive inhibitors bind with the enzyme, they cause a structural distortion in the enzyme, which diminish the catalytic power of the enzyme. Okay, and this graph shows the equation of reaction rate when uncompetitive inhibitor is added. Okay, so it's like this: V0 equal to Vmax substrate concentration divided Km plus alpha prime substrate concentration. Okay, and the value of alpha prime is equal to 1 plus concentration of inhibitor divided K, another constant. Okay, and this is the linearized form of the reaction rate, and if we plot this equation in the graph, it looks like this. Okay, okay, now another type of reversible inhibitors, the non-competitive inhibitors. Okay, and these kind of inhibitors, they interact with the enzyme in a way that affects both substrate binding and the catalytic activity. Okay, and this phenomenon is known as mixed inhibitions because they act as both like a competitive and uncompetitive inhibitors. Okay, so they have a mixed effect. They both interfere with the substrate binding and also with the catalytic activity of the enzyme. Okay, so that's why they are named as sometimes mixed inhibitor or non-competitive inhibitor. Okay, and presumably a mixed inhibitor binds to the enzyme sites that participate in both substrate binding and catalysis. Okay, and one example is metal ions. Okay, so metal ions which do not compete directly with substrate for binding to an enzyme active site as do competitive inhibitors, but may act as a mixed inhibitors. Okay, also mixed inhibitors can bind at a site distinct from the substrate active site. Okay, but it binds either to the free enzyme or the enzyme substrate complex. Competitive and inhibitors bind only with the free enzyme, and uncompetitive inhibitors, they bind only with the ES complex. Okay, but mixed inhibitor, they can bind either with the free enzyme or the EX complex. Okay, so thus the non-competitive inhibitor, they can hinder both the binding of substrate and decrease the turnover number of the enzyme, because as they interfere also, as they also interfere with the catalytic activity of the enzyme, they can decrease the turnover number of the enzyme. Okay, they have both effect. Okay, okay, and this slide, it shows the ways of uncompetitive inhibitor, how they works. The non-competitive inhibitor. The non-competitive inhibitor binds both with the free enzyme and also with the enzyme substrate complex. Okay, and they bind either in the active site of the enzyme, which act as both substrate binding and catalytic activity, and also they can bind in a distinct site other than that active site and can interfere with the catalytic activity of the enzyme. Okay, and this graph, this is the rate equation when non-competitive inhibitor is added with an enzymatic reaction, which is V0 equal to Vmax substrate concentration divided alpha Km plus alpha prime substrate concentration. Two new parameter comes here. Okay, and the value of alpha is 1 plus I divided Ki, and the value of alpha prime equal to 1 plus I divided K1 I prime. Okay, this is the linearized form of this equation, and this is the graph if we plot this equation in the graph. Okay, okay, now this slide, it shows side by these are side by side plotted graph of the competitive, uncompetitive, and mixed inhibitions. Okay, so we already saw these graphs in previous slides. Okay, this table summarize the effect of reversible inhibitors, different reversible inhibitors on the Michaelis-Menten reaction. So we already know that when inhibitors are added, the reaction equation, the reaction rate changed. That means the equation changed. Okay, so this table actually summarizes that what kind of effects are there. Okay, so like I mean the effects of inhibitors on Michaelis-Menten reactions. So for example, in case of competitive inhibitors, they increase the Km value, okay, and the uncompetitive inhibitors, they decrease the Km and Vmax, okay, and mixed or non-competitive inhibitors, they decrease the Vmax but may increase or decrease the Km, okay. So in case of competitive, Km increase. In case of uncompetitive, both Km and Vmax decrease, but in case of non-competitive, Vmax decrease but Km can either increase or decrease, okay. Okay, so that's all for this part. In the next part of this lecture, I'll talk about irreversible inhibitors. Okay.